Artemisinin, from ancient herbal remedy to a modern antimalarial revolution. The discovery of artemisinin stands among the most remarkable examples of translating traditional medicine into modern therapeutics. Since the introduction of quinine from the bark of the cinchona tree in the 16th century, no natural product has had a comparable impact on malaria treatment. While quinine remained the cornerstone of antimalarial therapy for centuries and was later supplemented by synthetic drugs such as chloroquine, the rapid emergence of multidrug-resistant Plasmodium falciparum during the 1960s created an urgent demand for new therapeutic agents. Artemisinin, a sesquiterpene lactone containing a unique 1,2,4-trioxane endoperoxide bridge isolated from Artemisia annua, sweet wormwood, fulfilled this need and transformed global malaria control. The medicinal use of Artemisia annua can be traced back more than 2 millennia in traditional Chinese medicine. Ancient medical texts, including the recipes for 52 diseases, 168 BCE, Zhou Houbei Jifang, 340 CE, and Ben Chao Gang Mu, 1596, described the use of Qinghao preparations for intermittent fevers, a hallmark of malaria. However, conventional hot water extraction produced inconsistent therapeutic outcomes because the active constituent is heat-sensitive. Modern discovery began in 1967 under China's secret military Project 523, initiated to identify new antimalarial drugs during the Vietnam War. Among the scientists involved, Professor Tu Youyou made the decisive breakthrough by carefully examining Ge Hong's 4th-century prescription, which recommended soaking rather than boiling the herb. This insight prompted the replacement of high-temperature ethanol extraction with low-boiling ethyl ether, preserving the active compound. In 1971, the optimized extract demonstrated complete parasite clearance in rodent malaria models, and in 1972, the active crystalline molecule, initially named Qinghaosu and later artemisinin, was isolated. Early clinical studies showed remarkable efficacy against both Plasmodium falciparum and Plasmodium vivax, including strains resistant to chloroquine. These pioneering discoveries ultimately earned Tu Youyou the 2015 Nobel Prize in Physiology or Medicine, although the award also highlighted the collective contributions of hundreds of scientists participating in Project 523. Structurally, artemisinin differs fundamentally from quinoline antimalarials. Its highly oxygenated sesquiterpene scaffold contains an unusual endoperoxide bridge essential for biological activity. Within malaria parasites, iron released during hemoglobin digestion cleaves this peroxide bond, generating highly reactive oxygen-centered and carbon-centered radicals that alkylate parasite proteins and membranes, leading to rapid parasite death. This unique mechanism minimizes cross-resistance with earlier antimalarial drugs and established artemisinin-based combination therapies, ACTs, as the World Health Organization's recommended first-line treatment for uncomplicated P. falciparum malaria. Despite its extraordinary efficacy, artemisinin presents formidable manufacturing challenges. The molecule contains multiple stereocenters and a chemically fragile peroxide bridge, making total chemical synthesis exceptionally lengthy, low-yielding, and economically impractical for commercial production. Consequently, the pharmaceutical supply has relied primarily on extraction from A. annua, where artemisinin typically accumulates at only 0.1 to 1.0% of the dry leaf weight and varies considerably with cultivar, geography, climate, and harvest time. These fluctuations have historically produced unstable supplies and volatile market prices, complicating global malaria treatment programs. Advances in biosynthesis and synthetic biology have largely addressed these limitations. Elucidation of the artemisinin biosynthetic pathway, including enzymes such as amorpha-4, 11-DNA synthase, ADS, CYP71, AV1, DBR2, and ALDH1, enabled metabolic engineering of microorganisms. A landmark achievement came through engineering Saccharomyces cerevisiae to ferment sugars into artemisinic acid, a late-stage biosynthetic precursor. Subsequent four-step chemical conversion of artemisinic acid produces semi-synthetic artemisinin, providing a scalable and reliable manufacturing platform independent of agricultural harvests. Commercial production of semi-synthetic artemisinin, pioneered through synthetic biology collaborations involving Amyris and industrial partners, has significantly improved supply chain resilience while reducing dependence on seasonal cultivation. Current research seeks to further increase artemisinin production through genetic improvement of A. annua, metabolic engineering, genome editing using CRISPR/Cas systems, optimization of glandular trichome development, and microbial cell factories capable of producing advanced intermediates or even the final molecule. Simultaneously, medicinal chemists continue to design fully synthetic peroxide antimalarials, including ozonide analogues such as arterolane, OZ277, and artifenamel, OZ439, inspired by artemisinin's unique pharmacophore. The discovery of artemisinin represents a landmark in medicinal chemistry, demonstrating how traditional medical knowledge, natural products, chemistry, pharmacology, and modern biotechnology can converge to address one of humanity's most devastating infectious diseases. Beyond saving millions of lives through artemisinin-based combination therapies, artemisinin has stimulated entirely new directions in antimalarial drug discovery and established synthetic biology as a practical solution for manufacturing complex natural products at industrial scale.